This lesson is about DNA replication. Here are your objectives. So just a reminder that DNA replication starts at origins of replication, and in prokaryotes there's one of those per chromosome. So the DNA gets opened up and forms a replication bubble. And at each end of the replication bubble, so here and here, we have a replication fork, and that's what I'm showing in larger here. So each replication bubble has two replication forks. We're going to look at the one on the left. The first enzyme to consider is helicase, and helicase is going to bind to the DNA and it's going to unwind it and separate the two strands. So here's helicase, and we're at the left replication fork, so helicase is moving in that direction. Now that unwinding creates more strain and tension in other parts of the DNA, and so another enzyme called topoisomerase is going to relieve that strain by creating nicks in the sugar phosphate backbone, letting the DNA unwind, and then putting it back together. The next enzyme we'll consider is primase. DNA polymerization needs a starting point, and primase makes primers that provide that starting point. So primase could attach here and will move in that direction. Now I should go back and label, let's make this the 5' end of this top template strand. That makes this the 3' end, and so because DNA is anti-parallel, that's the 3' end of this one and the 5' end here. So primase, shown here, is operating in a 5' to 3' direction in terms of the RNA polymer that it's leaving behind. It's attaching nucleotides on the 3' end. So here shown in green is a short RNA primer that the primase has made. And now DNA polymerase can attach, and it's also going to move in the 5' to 3' direction. And now here shown in blue, this is the matching DNA that has been made on this top template strand. Again, the DNA polymerase, like the primase, has gone in the 5' to 3' direction in terms of the strand that it is laying down. This top strand, where the DNA polymerase goes in the same direction as the helicase, which was shown here in green, this is the helicase. This top strand where the DNA polymerase is traveling in the same direction as the helicase is called the leading strand. And on the leading strand, we just see continuous movement of the DNA polymerase chasing after the helicase. On the bottom strand, we start in the same way, where we have a primase attach, and then it's going to move in a 5' to 3' direction in terms of the primer that it's making. And so here we see the RNA primer, and now DNA polymerase will attach again and extend that with a segment of DNA. However, on this bottom strand, we have a bit of a problem. Again, both the primase and the DNA polymerase are moving in the 5' to 3' direction, but the helicase is going away behind them and leaving open DNA track that needs to be filled. And so what happens there is that we have multiple primases attaching, so another primase would attach and make another primer, and then another DNA polymerase would attach and create more DNA. And so through this kind of backstitching method, we end up with multiple segments with a primer and then DNA following, primer and then DNA following. And so we need to get rid of these primers because they're RNA, not DNA, and then we need to join the whole thing together. The DNA polymerase we've been talking about so far, that's doing the main job of making most of the DNA, is called DNA polymerase 3 in bacteria. And I should say that what we're looking at here is DNA replication in bacteria. In eukaryotes, it's basically the same, but some of the enzymes have different names, and sometimes there's more varieties of them. But the overall idea is the same. So this was DNA polymerase 3 that we've been dealing with so far. DNA polymerase 1 now, on this bottom strand, which we call the lagging strand, DNA polymerase 1 is going to remove this primer and replace it with DNA. So we've replaced the primer, which was RNA, with DNA. There's still a nick in the sugar phosphate backbone that the DNA polymerase 1 can't close, and so another enzyme called ligase will fix that up and join the sugar phosphate backbone. And now we have a complete strand that is only DNA with no RNA. So on the leading strand, we saw that DNA polymerization went from 5' to 3' and was continuous, while on the lagging strand, while DNA polymerization still goes from 5' to 3', but it's discontinuous. And we have these RNA primers and then a segment of DNA, and then RNA primers and a segment of DNA, and then we end up having to join those together. These segments of DNA that are made between the RNA primers are called Okazaki fragments. And so we have multiple Okazaki fragments that are made on the lagging strand, and then they are joined together as DNA polymerase 1 removes the RNA primer, and then ligase joins the sugar phosphate backbone. And because it bears repeating one more time, the difference between the leading strand and the lagging strand is necessitated because of the fact that the DNA polymerase and the primase are always operating from the 5' to 3' direction. On the lagging strand, shown here on the bottom, that 5' to 3' direction takes them in the opposite direction from the helicase, leaving this open track behind it, and therefore the DNA needs to be made discontinuously in these Okazaki fragments that are joined together. Whereas on the leading strand here, shown on the top, the 5' to 3' direction that the polymerase operates in takes it in the direction chasing after the helicase, and so polymerization can happen continuously. And again, while this is illustrated in bacteria, in eukaryotes it's very similar, with some differences like the polymerases have some different names. Once again, the direction of polymerization on the leading strand and the lagging strand is both 5' to 3'. However, on the leading strand, the DNA polymerase moves in the same direction as the helicase, whereas on the lagging strand, it moves in the opposite direction. And that's what necessitated that discontinuous polymerization with the Okazaki fragments, whereas on the leading strand, polymerization was continuous. So that's a look at DNA replication.